You know, we're practically swimming in data these days, especially in biology. But with all that information floating around, how do we find the real story? But the real question is, how do we separate a meaningful signal from all that distracting and often misleading noise?
I mean, imagine you're a scientist. You're staring at data from over 20,000 genes all working inside a cell. So, how do you find the few that actually matter? That's the billion-dollar question, whether you're fighting cancer, trying to develop new drugs, or just trying to figure out the basics of how life works.
So that's what this explainer is all about. We're going to look at a really powerful statistical tool that helps scientists on this exact quest. We're going to dive into a method called DESeq2 and see how it turns messy, unreliable data into clear, trustworthy discoveries. All right, so to really get why the solution is so cool, we have to talk about the problem first. And, well, the problem is that raw genetic data, it can be really, really deceptive.
Okay, so the main tool scientists use here is called RNA sequencing, or just RNA-seq for short. Think of it like this. It's like you're taking a snapshot of a cell, running a massive census, really, to see which genes are on and at work under different conditions, like before and after a new drug treatment.
But, and this is a big but, these experiments can be super expensive and tricky. What that means is scientists are often working with just a handful of samples, maybe only 2 or 3 for each condition. And with so few data points, our measurements are just naturally full of uncertainty. And that uncertainty, it sets up this perfect trap. The data shows this really strong variance for genes with low activity or a low read count. So what does that actually mean? It means the stuff that looks like the biggest, most exciting change, these huge log-fold changes, often comes from the genes with the weakest, most unreliable data, not the ones with real biological importance. And that, right there, is a recipe for a scientific wild goose chase. It sends researchers chasing after signals that are basically statistical ghosts. So the real question, the crucial question, is how in the world do you tell what's real biology versus just random noise? Okay, so this is where things get really clever. A brilliant statistical idea comes in to save the day. It's the engine behind this method called DESeq2, and it's all built on this one simple concept: borrowing strength from the crowd. The technical name is empirical Bayes shrinkage. I know, it's a bit of a mouthful, but the idea is actually pretty simple. So instead of just taking one gene's noisy data at face value, what it does is it looks at all 20,000 genes to learn the overall trend. Then it uses that big picture view to gently nudge or shrink those really wild, unreliable numbers back towards something more sensible. And this process does 2 really important things. First, it tames that random variability, what scientists call dispersion. And second, it adjusts the effect size, basically how much the gene looks like it changed. It just pulls those crazy outliers back down to earth, toward a more believable reality. And you can see the difference right here. It's pretty dramatic. Look at the left. That's before shrinkage. The numbers are all over the place, totally exaggerated for the low-count genes. But then look at the right. After DESeq2 shrinkage, everything is so much more stable. You're getting a picture that's way more realistic and honestly trustworthy. Okay, so the idea sounds great, right? It's elegant, but does it actually hold up in the real world? Does this fancy statistics actually lead to better science? Well, the authors of the paper asked that exact question, and they came up with a really smart way to test it. They designed a little experiment to see if this whole shrinkage thing really could make science more consistent. So what they did was they took one big data set and just split it right down the middle. Then they analyzed each half like it was its own separate experiment. And the results? Well, without shrinkage, the 2 halves didn't agree very well at all. But with DESeq2's shrinkage, wow, the agreement was incredible. The error, the difference between the 2 experiments, dropped by more than 70%. But wait, it gets even better. This is the real kicker. They looked at the top 100 most changed genes from each half. Without shrinkage, you know how many genes were on both lists? Just 21. That's terrible. But with shrinkage, that number jumped to 81. 81! That's a night and day difference in reproducibility. So, what does this actually mean for science?
Why is this such a big deal? Well, it means researchers can finally be confident that they're chasing real biological leads, not just statistical ghosts. It means less wasted time, less wasted money, and ultimately, it just speeds up the whole process of discovery. And it doesn't stop there. This method isn't just for cleaning up lists of genes. It's also an amazing tool for seeing the big picture, for finding those hidden patterns in all that messy data. You see, if you just look at the raw data, all that noise can really throw off the algorithms that are supposed to find patterns. But DESeq2 has this special tool, a transformation they call a regularised log, or rlog. By stabilizing all that variance, it basically tidies up the data, so tools that group samples can finally see the true biological story and get it right.
And the central idea, taming noise by borrowing strength from the crowd, it's just so powerful and fundamental that it's exploded way beyond just RNA-seq. Now you see it being used as a key method in all sorts of fields, like chromatin assays, metagenomics, even in those super cool CRISPR gene editing studies. All right, so let's tie this all together. The bottom line is this. Raw biological data, especially from just a few samples, is super noisy. And those big flashy effects you see at first, they're often just statistical illusions. The absolute genius of DESeq2's shrinkage is that it gives us a rock-solid, statistically sound way to cut through that noise. It leads to science that's more stable, more reliable, and frankly, more true. So back to our big question. In a world just drowning in data, how do we find what's true? Well, DESeq2 gives us a pretty powerful answer. It's a great reminder that sometimes the clearest signal isn't the one that's shouting the loudest. It's the one you hear when you listen carefully to the wisdom of the whole crowd.